Yeah. Hi, everyone. So Dr. Akiko Nishiyama obtained her MD at Nippon Medical School, then further pursued her PhD at Nagata University in Japan. She is now a professor of physiology and neurobiology at the University of Connecticut. But Dr. Nishiyama has published over 100 research articles. Her research focuses on molecular and cellular mechanisms of heterogeneity and lineage plasticity of NG2 glial cells and neuroglial interactions in both normal development and in lesion repair. The techniques used in her lab include mouse genetics combined with immunohistochemistry, tissue culture, both fluorescence and confocal microscopy, and biochemical and molecular biological techniques, including RNA sequencing and chip sequencing. So I just want to say thank you so much for coming to speak to us today, Dr. Nishiyama. We're all really excited to listen to and discuss your research.
Okay, thank you so much, Juliana. It was a great comprehensive introduction, and it's very nice. And I'm sorry, it's been probably a little while since you read the papers, and I had to reschedule this, but I really appreciate the invitation from Dr. Raskin, and it's a great honor for me to talk to you. And so I think I'll go ahead and share my screen. Let's see, it seems like the screen share is disabled.
Try now, it should be okay.
Okay.
It's not letting you?
Oh, now it's okay. So maybe I'll just do the PowerPoint. Can everybody see this?
Yes, absolutely.
Okay,
great.
So please feel free to interrupt me anytime when you have questions, and don't feel shy about asking questions, because it's just fun to let the discussion go where your interests are. So what I like to talk about today is just an introduction to what these glial cells that we've stumbled across many years ago might be doing in the brain. And like most research topics, we don't have a complete answer to this question, but I thought I'd share with you some of the paths we have taken to try to understand what these cells do. And if I remember from my frustrations through the my undergraduate career, I was very interested in learning about how things work, but I had a hard time understanding how to go about researching things. And one of the things that I found very difficult is to try to understand how you can ask or answer questions with specific techniques. And as undergraduate students weren't exposed as much to various techniques. So how we go about answering questions was a very obscure and rather mysterious process. So I'd like to try to weave in some of the approaches we've taken to solve some of the questions. So just to start, you are probably all very familiar with the cellular composition of the brain, but I just want to throw this question at you and ask you, how many different kinds of cells do you see here? Anybody brave enough to jump in? Just unmute yourself and so this is a cartoon of the central nervous system. It could be the brain or the spinal cord or the cerebellum.
Our principal students should know the answer to this.
So should the current issues in brain and behavior.
What are these big cells here? It's a little bit hard to get used to this cartoon, but this is a big cell, the cell body here, and some long thing coming down from here.
A neuron?
Yes. Okay, so this is the key player in the central nervous system. Neuron is the cell that fires action potentials, and they can transmit information. But what are all these other little things here? Have you heard of these things?
We
Are they?
astrocytes.
Yeah, so these are astrocytes. These have, they're called astrocytes because they look like stars. You know, they have these processes. And the tips of these processes contact many different structures. This is a blood vessel, and it's contributing its end feet to the formation of the blood-brain barrier. Astrocytes also wrap around synapses. So I think these are some of the synapses at neuron synapses, but astrocytes also are involved in glutamate uptake and important functions at neuron synapses. Anybody else have any idea of what these other cells are? Do you know what this cell is? That seems to be connected to the axon via this kind of a tube.
Are they oligodendrocyte?
Yes, very good. So this is an oligodendrocyte, and it makes these fatty sheets that are called myelin. And myelin, you might have heard, is an insulating, has an insulating function around the axon. So it speeds up the conduction, and it allows axons to conduct in a saltatory manner from the node around it next. So we're different from the squid. We're much bigger in size, and so our motor neurons have to transmit signal to the spinal cord neurons that could be 3 feet, 1 meter in distance. And if we transmitted signals at the speed that squid axons transmit, we would have to wait forever to get our leg muscles to move and so on. So that serves the purpose of speeding up the conduction velocity in the nervous system of larger animals. And then we have these mesenchymally derived cells that are microglia, and they play an important role in surveillance of the nervous system in inflammatory responses to keep the homeostasis in the brain. And then you have these ependymal cells that are lining the ventricles. So you have all these cell types. And then this cartoon is shading the upper portion of this picture a little bit brownish color. And this part is white. And this is because we anatomically we separate the gray matter from the white matter. And the gray matter is called gray because it looks gray to the naked eye. And the white matter we call white matter because it looks white and shiny. And it looks white because the myelin here, the fatty sheets of the myelinating oligodendrocytes that surround the axons give it a translucent, shiny white appearance. So you can distinguish the white and the gray matter from the by naked eye. But then how do we know that these cells exist in the central nervous system? So this is the question that had puzzled anatomists more than 100 years ago. And just as an example, this is a human brain cut along the midline, and here's the cortex. Here's a sagittal section of the brainstem. And you can see some whitish area that corresponds to the white matter, and some grayish area where the tissue is rich in neuronal cell bodies, but not axons. And it's quite clear if you cut a sagittal section through the cerebellum, you can see the white matter going through the core of the cerebellum. It appears shiny white compared to the grayish area which represents an area rich in cell bodies. This is an area rich in axons. So how do we go about identifying cells in the brain? And these are just a series of pictures showing how one can look at Purkinje cells in the cerebellum and some very crude analysis of the cerebellum. There's a histochemical stain called hematoxylin and eosin will give you a picture like this. So this is the cerebellar cortex, and you can see the white matter here, and some purple is a stain for nuclei. So it represents individual cells. And this is too low a mag to look at cells, but you can see this layer here that's dark purple is rich in granule cells. So this is a cluster of one kind of neurons that's located here. And then you have a rather cell sparse area called the molecular layer of the cerebellum. And then you have this row of large cells that are that we know as Purkinje cells. And these cells were discovered by a Czech anatomist called Jan Purkinje. And he looked at these kinds of chemical stains, and he was able to make this kind of a drawing and said, these are cells that look very different from the other cells. They must be some special neuronal population. And there was a lot of speculation back then because you couldn't really do functional experiments right away. And so these are some other histochemical stains. And then came an Italian anatomist called Camillo Golgi. And this is, so Purkinje saw these cells in mid-1830s, and Golgi was working 40, 50 years later. He developed the silver nitrate staining method. So he applied this chemical stain to sections, and he was able to see this kind of a picture on the same kind of tissue as these. But what's really different is that you get occasional cells labeled, not all cells, but each cell is labeled in its entirety. So you can label all the dendrites and a bit of the axon here, and a much to a much greater extent than what one could with earlier histochemical stains. So this gave us an idea of what the Purkinje cell really looks like. It's almost like an elephant touching, or 6 people touching different parts of an elephant and calling the elephant a long tube if you touch the trunk, and calling it a flat surface if you touch the body, and so on. So depending on what kind of stain you apply to the sections, you get a completely different view. And 100 years later, we now apply a technique called immunohistochemistry to reveal some proteins that are present in specific populations of neurons or glial cells, or different cell types, different structures in the tissue. And so this is an immunohistochemical stain for a protein called calbindin, which is typically expressed in Purkinje cells, but not limited to Purkinje cells. So you can see the soma of the Purkinje cells and the dendrites here. And then in addition to immunohistochemistry, one can see Purkinje cells really nicely by a more genetic approach, which just expresses GFP, green fluorescent protein, with a promoter, which is a piece of DNA that drives the expression of its downstream target. So you put a Purkinje cell promoter next to the GFP gene, and you can get this kind of a nice labeling of Purkinje cells and their axons in the cerebellum. So this just gives you a flavor of all the different ways in which one can visualize different cells in the brain. And so following this example of neurons, what did we do to study oligodendrocyte precursor cells, or NG2 cells? And we have a molecule that seems to be very specifically expressed in these cells, and this is called the NG2 molecule. This was what really started the research for OPCs. I'm going to call the oligodendrocyte precursor cells OPCs, short. And this molecule was discovered by a scientist called Bill Stalcup in La Jolla, San Diego. And he was he made a lot of monoclonal antibodies against different cell surface proteins of cells. He grouped some, he had some cell lines. These are immortalized cells from the nervous system. And he grouped these cells, neuronal or non-neuronal, depending on whether they could generate action potentials or had ion channels. And there was a group of cells that just he couldn't be classified as purely neuronal or glia because they didn't generate action potentials, but yet they had sodium and potassium ion channels. And so back in the 70s, that was a mysterious group of cells that he didn't know what they were. And so he identified a protein specifically expressed by these cells, and he called them NG2 because they were neither neuronal nor purely glial. And that was the start of the studies. And I was involved in identifying the structure of the primary sequence of the NG2 protein so that we could generate reagents with them. And by looking at the amino acid sequence of this protein, we could predict that this protein is a transmembrane protein with a big part of it sticking out into the extracellular portion of the plasma membrane. And when we apply immunohistochemistry using an antibody to this NG2 molecule, this is what we see. So this is an adult rat brain stained for NG2. You can see the dark product is the reaction product. So you can see the cell, very small cell body, but very elaborate processes, extensions from the cell bodies. And these cells are everywhere. This is a low mag picture showing a very even distribution of NG2 expressing cells. Or we didn't know back then, so they were oligodendrocytes, so I'm just gonna call them NG2 cells. And this is the cortex. This is the corpus callosum magnified here, this bit of the hippocampus. But these cells are everywhere throughout the adult brain. So the main question was, what are these cells? And that was a puzzle in the late 80s. And so I had finished my medical degree in mid 80s, and I went to, wanted to understand how glial cells interact with neurons. And I went to, first started as a resident in the pathology department because I thought maybe if I looked at neurodegenerative diseases, I would understand a little bit more about how different cells were involved in different degenerative processes. But then it turned out that pathology, you look at autopsy material, you look at the aftermath of all the events that occur. So it was really difficult to dissect what goes on. So I decided to switch my training, get more training in molecular cell biology, and try to do some more basic experiments to test the role of glial cells and neuronal function. And so this was an ideal lab for me to go and get trained after my graduate studies, because he had just identified these cells. He didn't know what they were. They didn't look like astrocytes. So they looked like an interesting population of glial cells that I could go and study. And so that's what led me to seek my training with him. And so the main question we had back in the 90s was, what are these cells? Nobody knew. There was one old paper from 1900 by a Scottish scientist called William Ford Robertson. And he had drawn a picture of cells that looked just like these. And unfortunately, though, he mixed them up with microglia, and he called them, he lumped microglia and these cells together. So researchers that came after him just couldn't figure out what he was talking about. So his work had gotten buried. But since then, there is a gap of about 70 years before these cells were brought to the attention of researchers. And they, because of the lack of identification methods, people didn't know that these cells existed. And suddenly we came across these cells that were very, very abundant in the brain. We didn't know what they were. So we had to start figuring out how to go about solving the question of what these cells are. So first we did the obvious thing of comparing the immunohistochemical reaction of these, the NG2 protein with other known markers for different cell types. So astrocytes are known to express a protein called glial fibrillary acidic protein, shown in green here. And if you look at, so you can do immunofluorescence staining, double labeling, so you can compare the expression of two proteins and in the tissue section. And here you have the red NG2 expressing cells and the green cells, and they don't seem to overlap. So NG2 cells don't appear to be astrocytes. Neither are they microglia. So here again, you have a different marker for microglia. And these are the red NG2 cells and the microglia. You have cell body of NG2 cells here and cell body of microglia here. They are not the same. So the other cell type we talked about before are the mature oligodendrocytes. And these are shown in purple here. Sorry, I got the colors mixed up. So the purple is the oligodendrocyte, and the green, it's reversed, is the PLP, which is marker for mature oligodendrocyte. And there's no overlap. So these comparisons suggested that NG2 glia are not distinct from any of these cells that we knew about. So we had to rewrite the textbook and say that, well, there's another population of cells in the brain that we didn't know about. And these can be identified by the expression of NG2, and we've got to figure out what they are. So how do we go about doing this? Well, one clue that had been generated before I started working on these cells, it came from cell culture studies. So for these studies, you dissociate cells from a young mouse or rat brain, and you watch the cells differentiate over time. So you can isolate cells that express NG2 from the optic nerve or the brain or the cerebellum, spinal cord, and then you can watch them differentiate, turn into other cell types over time in culture. And you can see that these green cells are now starting to be stained by this red marker, which is an oligodendrocyte marker called galactocerebrocyte. And then eventually you get this nicely differentiated oligodendrocytes. So by catching this transition in culture, Stalkup and colleagues were able to conclude that, at least in culture, these cells can turn into oligodendrocytes. So now the challenge was to see if we can show this in vivo, in the tissue. And how do we go about doing that? That's turning into quite a challenge. But at least in culture, one could predict that these immature cells, so NG2 expressing cells, are likely to be these immature cells that can then differentiate into oligodendrocytes that are known to turn into myelinating cells. And some of the markers that we use, these NG2 is a marker for the dividing progenitor cells. And then once they differentiate, they no longer divide. We call this, they become post-mitotic. And then these downregulate, lose the expression of NG2, and then they acquire the expression of markers that are typically expressed on mature oligodendrocytes. We looked at PLP. CC1 is another mature oligodendrocyte marker that we're going to be looking at quite a lot. So just remember this. And so the big question, puzzle, or some conundrum that we had to solve was, how can we demonstrate that these cells turn into oligodendrocytes in vivo? And because NG2 is downregulated before these cells turn into other cell types, we couldn't just rely on simple immunohistochemistry, because we would have oligodendrocytes that don't have NG2. So we don't know where the oligodendrocytes came from. And then NG2 itself is downregulated before these cells turn into other cell types. So we wouldn't know what these cells turn into. So we had to come up with a genetic approach. And this requires the use of the Krelok's recombination technology. And I'm not sure if people are familiar with that. I'm just going to explain it briefly here. So this requires the use of 2 transgenic mouse lines. And by combining them, you permanently express a fluorescent marker specifically in all the progeny of NG2 expressing cells so that you can follow the fate of these cells. So here's the first mouse that expresses Cre recombinase under the regulatory control of the NG2 gene. And the second mouse expresses yellow fluorescent protein. It's almost like green fluorescent protein under the control of the constitutive active promoter. But normally YFP is not expressed because there's a transcriptional stop signal here. And when you combine these 2 mice, Cre is a recombinase, and it excises any DNA sequences that are sandwiched between these 2 Cre target sequences called LOXP. LOXP is a short 34 base pair sequence. Anything that's sandwiched in between 2 LOXP sites gets removed. So ultimately, wherever you have Cre, which is all cells expressing NG2, there's recombination that goes on. You remove the stop and you permanently activate expression of YFP. And YFP is now under the control of a constitutively active promoter. So you can get YFP expression in any progeny as long as this cell type came from an NG2 expressing cell type. So the expression of YFP is not dependent on the expression of NG2. And so one can imagine these kinds of cell populations. So in cells that are expressing NG2, recombination allows the onset of YFP expression. So you get red immunohistochemically labeled NG2 expressing cells that are also green. But then when the NG2 expressing cells go on to develop into some other cell type that no longer expresses NG2, it loses the NG2 and it becomes green only. So one can see examples of this. So here's a green cell that's also red. NG2 is a cell surface protein, so you can see the contour of the cell, not the cytosol. And this red cell is YFP positive. But then you have green cells that typically have the morphology of a typical oligodendrocyte. And these cells are green only, and they're not labeled with the NG2 antibody. So one can see that they have already downregulated NG2 expression, and they've become something else. And in this case, they have become oligodendrocytes. So this was really the first demonstration that we wrote and convince people that, yes, the NG2 expressing cells are precursors to oligodendrocytes. And up until these results came in the mid-2000s, there was quite a bit of confusion in the field. People didn't know what NG2 cells were. There was some speculation that these are stem cells that could give rise to all kinds of cells, neurons, astrocytes, and oligodendrocytes. Others said these are microglia. They look like microglia. So this put to rest a lot of the questions in the field. And one could now go on to do the next set of experiments, assuming that these are oligodendrocyte precursor cells. So are there any questions so far? Anybody, is everybody okay with the Screlox technology?
I don't see any in the chat. This is great. You've been very clear.
Okay, I'll go on. So the next question was, we saw a lot of these cells in the adult brain. And when one thinks of precursor cells, one usually thinks of an immature brain, right? Precursors are important during development and they generate mature cell types. But then why do you have so many NG2 expressing cells in the adult brain? So that was a big puzzle that nobody knew the answer to. So we wanted to know if in the adult brain, whether the NG2 expressing cells could also give rise to oligodendrocytes. And you use the same approach, you get a little bit fancy. So instead of using constitutively active Cre, we use a Cre with an estrogen receptor-like sequence bound to it so that we can control the onset of the activation of Cre with a ligand called tamoxifen. And the reporter is exactly the same. And so when we combine these two mice, unlike the constitutive Cre, you have to inject tamoxifen to induce this recombination. So the beauty of this is that you can inject tamoxifen at a very young age in the perinatal period or in the adult mice. And then you can analyze the oligodendrocytes and find out how many of these oligodendrocytes are not colored green or whether they're green. And the green oligodendrocytes must have come from recombined NG2 cells that were NG2 positive OPCs at the time of tamoxifen injection. And just take one time point, for example, look at 60 days after Cre induction. You can assess how many of the oligodendrocytes have become green. And if you look at this data here, if you compare, so this bar means that when you inject tamoxifen at postnatal day 2 in the mice, about 75% of the oligodendrocytes have become green by 60 days after tamoxifen. So during the 60 days that have elapsed after tamoxifen injection, oligodendrocytes, 70% of the oligodendrocytes have been derived from cells that were precursor cells. So there's quite a bit of a turnover. And this rate is slower in the adult, but still, nevertheless, you have about 50% of the oligodendrocytes being generated from precursor cells during the 60-day period. And the rate of differentiation is a bit slower in the cortex, but they do differentiate. So now we have this assay. So we can start to ask some questions that are related to how might the differentiation of oligodendrocytes be regulated. And I'm just going to skip some of this. So now I want to talk about some of the work that was described in the Nature Neuroscience paper from 2014. And this is all the work that a former graduate of Trinity College, Bobby Hill, did while he was a graduate student in my lab. So we used the same technique. Bobby used the same technique, but he just added another assay, which is EDU incorporation. EDU is a thymidine analog, and it is preferentially incorporated into dividing cells. So you can not only label progenitor cells by marking them with YFP using the Crelox technology, but you can also label dividing progenitor cells. And when they differentiate into oligodendrocytes, they no longer divide. So any YFP positive cell that has taken up EDU must have been a progenitor cell, a precursor cell at the time of EDU injection. And you can see an example of this. So at 1 day, so you inject tamoxifen at postnatal day 8 and or 6 to 8 and induce Cre recombination, so induce YFP expression. Inject the EDU at P8. So 1 day after EDU injection, you see a doublet, which probably came from a single cell that divided into 2 cells. And most cases, these 2 cells express the progenitor marker NG2. But if you wait 4 days, one of these 2 doublets often has become a CC1 positive oligodendrocyte. So you can trace the fate of divided progenitor cells this way. And when you plot this data, you get a graph that looks like this. It's a bit complicated. The white bars represent the percentage of all YFP EDU positive cells that have become oligodendrocytes in white matter. And the black bars represent cells that have become oligodendrocytes in the gray matter. And you can see that with time, you see a gradual increase in the percentage of YFP positive divided cells that have become oligodendrocytes. But this plateaus at about 3 days after the EDU injection. So that suggested, and this happens both in the white matter and gray matter. So that suggested that there's a very finite temporal window during which divided OPCs become post-mitotic and differentiate into oligodendrocytes. And this provides a very important regulatory period when the divided cells are susceptible to external influences that might influence their outcome. So if you need more oligodendrocytes, there might be something going on, signaling that would push these cells to turn into oligodendrocytes and survive better. So we looked at some of the external influences here, and Bobby did some slice cultures, and these you can manipulate the external conditions more easily than in an intact animal. And you take, it's a similar kind of a reporter Cree combination, and you watch the cells change over time. And what he saw was that when he takes slices from a P8 animal, and he did an experiment where he demyelinated the axons. So he got rid of oligodendrocytes, he got rid of myelin, and watched to see what happens. And so when he did that, this is a bit of a complicated experiment, but normally during the, so he did time-lapse recording, and normally during the about 50, 70 hour observation period, very few cells differentiate into oligodendrocytes. But if you create a myelin damage, then you can see that these cells that have divided now turn on the mature oligodendrocyte marker much more quickly, and they become oligodendrocytes faster. So if one were to plot this on top of this normal graph, the curve is shifted to the left. So if you create myelin damage, you allow the oligodendrocytes to differentiate much faster, much sooner from the divided OPCs. So the next we wanted to ask whether there's some influence by neuronal activity, because these cells myelinate axons. So there must be some dependence on neuronal activity. And does that matter? Does that affect the way OPCs differentiated into the oligodendrocytes? So Bobby did this experiment. It's well known that whiskers provide sensory input to the somatosensory cortex. And each whisker is very accurately projected to a specific region in the barrel cortex, in the contralateral somatosensory cortex. So you can map each whisker onto a specific region. And these NG2 expressing cells are very abundant in the somatosensory cortex, like everywhere else. So he asked the question of what happens if you clip whiskers from one side of the mouse and see if that would affect oligodendrocyte differentiation from proliferating, dividing OPCs. And so I'm going to go through some graphs. So it's the same experiment. He did tamoxifen injection to induce Cree, and then injected EDU either at P6 or P8, it doesn't matter. And then he waited a certain number of days, 4 or 6 days, and counted the number of oligodendrocytes. So the total number of oligodendrocytes was slightly decreased in the deprived contralateral cortex, but not in ipsilateral uninvolved cortex. And if you look at the YFP positive CC1 positive cells, which represent newly developed oligodendrocytes that had become YFP positive at the time of tamoxifen injection, there's a greater drop. And on top of that, if you look at the proliferating OPCs that turned into oligodendrocytes, it was very dramatically reduced in the deprived cortex, but not in the normal cortex. And he did all these experiments in the motor cortex and there was no change. So there was a very specific defect in oligodendrocyte differentiation from divided OPCs that are supposed to become post-mitotic and turn into oligodendrocytes. So this was quite exciting for us, and he did some additional experiments to show that there's increased cell death in the involved cortex, in the affected cortex. So it seems like if you deprive the sensory input, you compromise the survival of newly differentiated oligodendrocytes that have that become post-mitotic and turn into oligodendrocytes. So, and that would affect the degree of myelination and the circuit function later on. So this is just a summary of what this data suggests. So OPCs are dividing progenitor cells, and myelin damage accelerates their differentiation into oligodendrocytes. If you deprive them of sensory input, you compromise their survival and you get fewer cells differentiating into myelinating oligodendrocytes. So just lastly, I want to say, why is this really, why do you care about this? Why is it important? And there's a huge body of, growing field that's called, that studies myelin plasticity. And we're now learning that there's continued generation of myelin or myelinating oligodendrocytes from precursor cells throughout the life of the animal. And the production of new oligodendrocytes is affected by learning and memory. So if you block oligodendrocyte differentiation from precursor cells, the animals show compromised learning of new motor skills. They have compromised fear conditioning responses. And lastly, I just wanted to point out some exciting finding made by Tomasi and Paula Arlota several years ago that suggests that these are pyramidal neurons in the cortex. We think of myelin as segments of myelinated axon having segments of myelin sheaths all along, but that's not the case. And when they did 3D EM reconstruction of myelinated axons, you have gaps. So here, this is myelinated, but this segment is not. This is myelinated, but this is not. And this axon, this is largely myelinated. So there's a lot of heterogeneity in myelination of big pyramidal neurons that we used to think were all myelinated. So that makes sense. There's a lot of plasticity in myelination that seems to be very closely tied with learning and memory. And learning and memory affects the degree of myelination. And myelination also affects how one can learn, how effective learning and memory occur, how effectively those processes occur. So this is just to tie in with some context and circuit function. And so I think I'm pretty much, I'm going to end here, I'm going to skip. So we're still very interested in understanding some mechanisms of neuron glial interaction and how neuronal activity, how specifically neuronal activity influences oligodendrocytes. So this is just to show you the UConn campus and highlighting Bobby, who's the main player in what I've talked to you about today. And just lastly, if anybody is interested in UConn, we have a research day, usually in the fall. So we'd love for you to come and visit our campus with your faculty if anybody has some free time, and do by all means contact us if you're interested in connecting with us. And these are just some big groups of people who are studying different things in our department. So I talked all the time, but if you have any questions, please feel free to.
You have a question in the chat from Professor Seraphin. Can you speculate on how oligodendrocyte differentiation might be impacted by rearing in social isolation?
Oh, that's an interesting question. There's actually a couple of papers that suggest that reared animals in isolation versus in normal social context, and there's a significant defect in the degree of myelination, and that seems to be to involve some epigenetic marking defects.
Interesting, really interesting. All right, so I saw Jeffrey's hand and then Elena.
Awesome. Thank you so much for the talk. And so my question, I do developmental biology research where I use RNA interference to understand gene function and cell behaviors. And so I was just wondering if it's even possible to silence glial cells, and would silencing these NG2 glial cells via, say, CRISPR-Cas9 or RNA interference would provide us any insight to its functional properties?
Yeah, that's a great question too. So it's RNA interference is a bit difficult to apply in vivo, but there have been other techniques that have been used to try to ablate these OPCs. And you do that. When you do that, the problem with that is that they really come back quickly. So there's a big, strong proliferative response. But there's one very interesting study that was done by a Canadian group a few years ago. And when they ablated NG2 cells, OPCs, in the hypothalamus, there was a big feeding behavior problem. And the leptin, there was a big problem with the leptin expressing receptors expressing neurons, and the mice got really fat. So they have to be doing something with these leptin receptor neurons, and we'd love to know what they do. And that kind of a CRISPR approach would be the way to go. I think somebody else, Elena.
Hi, yeah, thank you so much. I'm normally more interested in clinical cognitive, but I found your presentation incredibly interesting. So I just had a question. You had a paper in 2016 where you close out with the notion that further investigation would be required to determine the proportion of NG2 cells that become oligodendrocytes, and if there are NG2 cells that lose this ability permanently.
Mm-hmm.
And so I was wondering, because I know that article is a few years old, if you have continued with that research specifically and what those findings were.
Yeah, unfortunately, we don't have much new data since then, and it's been a long-standing question. We have all these NG2 expressing cells, and we can tell whether one of them turns into an oligodendrocyte, but we cannot really say how many of them really turn into oligodendrocytes. And if they don't turn into oligodendrocytes, what might they be doing? And that's a big open question that we still haven't been able to solve.
Okay, thank you.
You have a question in the chat. How significant do you believe it will be to develop findings on mitotic indices of OPCs for the purpose of understanding how to better treat and/or prevent multiple sclerosis?
So this is a big research area that's going on very actively, and people are really focusing on what really drives the oligodendrocytes or OPCs to become oligodendrocytes, and how do you get them to expand enough to generate enough oligodendrocytes? And so there are a number of different avenues that are being explored. I can't go into much detail here, but I'd be happy to discuss them with you if you want to send me an email.
That's great. What an offer. Thank you. Do different populations of NG2 glia exist within the same regions?
That's also a very hot topic, and people have looked and looked for because they arise from different locations, and people thought maybe different NG2 cells coming from different locations might have different functions. The only evidence that there might be some difference is that certain NG2 cells coming from a specific location have a preference for myelinating specific populations of axons and not the other types of axons. How that's regulated, I don't know. And there is some growing evidence that suggests that those that arise early may not be as capable of dividing as many times to repair a myelin defect. So the ones that come on later may have, this is just my speculation, may have undergone fewer divisions, and they may be sort of mitotically younger and are more plastic in terms of their ability to repair lesions. So that's an area of future investigation.
Oh, interesting. All right, here's a long one from Amy. There's a part in one of your articles where it mentions that it's a bit unclear whether the inflammatory responses in the spinal cord of adult rodents were caused by structural differences in the vascular and meningeal barriers, or that there were intrinsic differences in the oligodendrocytes and myelin of the spinal cord that make them more susceptible to immune-mediated damage. Between these two possible outcomes of structural differences versus intrinsic differences, which one do you think is more likely and why?
Inflammatory response. I think you must be talking about in the context of a spinal cord injury or some kind of a damage response.
Amy, can you clarify? Amy, are you there?
Maybe there's so there are differences in the way the brain cells and the spinal cord cells respond. I'm not sure if that's what you're referring to.
Yeah.
So some in the spinal cord particularly, I think there is a lot of meningeal response, fibrotic response to injury compared to brain. And maybe that's because of the proximity of the white matter to the meningeal surface and the spinal cord, but there's a lot of fibrotic response. And that might change the way in which the oligodendrocyte precursor cells respond. Usually those fibrotic responses, inflammatory changes, are inhibitory to oligodendrocyte differentiation and myelination. So that's a serious problem in spinal cord injury.
Great. So, any other questions from anyone? Obviously, our class should stay. Vivian?
Yeah, thank you for a great talk. So we know that oligodendrocyte, if it's damaged, there will be some effect. But do you know what will happen if there's an excessive amount of oligodendrocyte are generated? And has there been any studies about the communication between differentiated oligodendrocyte and the OPC?
Yes, and that's what we're really interested in. Stan, so when you artificially increase the number of OPCs, they differentiate and you get more oligodendrocytes. But there's a very, very tight regulation of the density of oligodendrocytes, much more so than the OPCs. So excessive oligodendrocytes that are generated die, and they don't seem to survive. We don't know what the mechanism is. We're now exploring the possibility that perhaps microglial cells function as sensors of the change in density, and they provide signals to promote OPC proliferation or just tell them not to proliferate more, or just lead to oligodendrocyte death. But that's still a very wide open area. But that's a very important question in terms of regulation and MS lesion repair and things like that.
Rachel?
Hi, thank you so much for your talk. It was really interesting. I'm wondering how you first got interested in neurobiology and specifically glial cells.
yeah, so what really piqued my interest was, I got trained in Japan, and in Japan, you decide to go into medical school at 18, right after high school. And I really wanted to do basic science, but this is not a good answer, but it was easier for me to go to medical school than to go to a very strict PhD program. So I went straight to a 6-year medical program. And I was always interested in basic science. And what intrigued me was a lecture in neurology, where we learned about all these neurodegenerative diseases. Neurons degenerate and they die, but nobody knew back in the 80s why they died. And we also learned in cell biology about glial cells, and they provide some supporting role for neurons. And I thought it'd be really interesting to find out if glial cells provided some important trophic function to the neurons. And that's when I decided that I really wanted to study cellular functions of glial cells, and especially in the context of neuroglial biology. But when I went to PhD, take my PhD exam, the interviewer said, what do you want to study? So that was a little bit off the topic back in those days, but I think it's still a very actively researched area now, and still a growing field.
Oh, wait, there's one more. What direction do you see your research going? What are you most excited about in terms of your research right now?
So right now, I think that there are two things that we're actively working on. One is the role of microglia in sensing and maintaining oligodendrocyte homeostasis, density homeostasis that I alluded to. The other is possibility of cross communication between neurons and OPCs. So what do OPCs secrete to influence neurons? What mechanisms, what signaling do the neurons use to tell the OPCs to differentiate or survive? And how do OPCs respond back to neurons? So that's an area that we're actively researching.
Anything else from anyone? So I want to ask, so I don't know if you all caught that she was talking about a Trinity College student who I actually distinctly remember having in brain and behavior, who's now assistant professor at Dartmouth University. He actually gave a talk on his research, which is incredible, about a year ago. So, so some of the work that she presented was actually the work of, he was then Bobby Hill, now he's Robert Hill. So I want to know how Bobby ended up in your lab as a Trinity student. Like, what was that? Did he apply for something post-graduation or while he was in grad school?
To come to my lab. And
yeah.
So we do graduate recruitment in February, and Bobby just applied, and he was among the pile of applicants. And I remember very well, he sat in my office. He's a very quiet fellow, and he still is. He said, so I said, why do you want to come to graduate school? What do you want to do in the future? And he said, I'm interested in research, but I don't know if this is the right place for me. I don't really want to teach. And so he said, and oftentimes quiet people are not are afraid of teaching. So I thought, maybe, don't worry about teaching. Just it seems like you've been enjoying research at Trinity, so why don't you get involved in research here, and then teaching will follow. And once you become comfortable with research, teaching is something that naturally follows, and it's something that you don't. You might be afraid of teaching initially, but eventually you'll learn to like it, and it ties in with your research too. And he just was just so talented in research. He was a tinkerer from the beginning, and I met his father, and he's also just like a replica of Bobby. And so they like to, they went in the microscope and they played around for the whole afternoon after this is defense. But so he liked to play around with things. If you're curious and you want to just understand how things work, research, I think I would encourage people to just get involved in research and don't be afraid of other things that you hear about, like you have to teach, you have to get grants. Those things just will fall naturally. Follow if you're you get a good research program going, and if you like research, I think it's not that hard to get a research program going. So that's how Bobby and I started in our interaction, and he just said, "Okay, I can come to your lab," and he had so I put him on different projects initially because we didn't have a well-developed project. So he suffered from that. He played around with a lot of different things, and ultimately he landed on his thesis project fairly late, about 3 or 4 years into his graduate work. So he persevered, but once he got going, he became really good at getting things in the right direction.
Fantastic. All right. If there's no more questions, then just thank you so much. We really enjoyed it. Appreciate
Thank you so much for the opportunity to speak.
Yeah, absolutely. And we'll definitely be, I'll be keeping track of the research day at UConn and make sure
I'll try to update you on that.
Great. All right, thank you.
Okay, thank you for joining us today.
Bye.
Bye.
So my class should stay on here because we're going to do the student presentations now. So why don't we take a little 5-minute bio break and then come back and we have the presentations. All right.